Hello, everyone. It is actually my great pleasure to introduce Máire today to speak about pre-cancerous evolution. So, Máire is currently a professor at Trinity College Dublin, and before that, where I met her, she was working with David Wedge, and she was also a postdoctoral researcher at Trinity College, not Trinity College Dublin, but Trinity College in Oxford, where she was working on cancer evolution. And then before that, she did a PhD in Cambridge on transmissible cancers. So I definitely recommend looking out that publication. It was very, very interesting. And yeah, today she's going to talk about pre-cancerous evolution, and I'm super excited. So, the floor is yours, please.
Cool. Okay, thanks to the organizers for giving me a chance to speak. And thanks everyone for hanging around until the very last talk at the end of the NGSymposium. I was supposed to give a talk in 2020 at the NGSymposium, but then that thing happened, and here we are now. So at the time I was supposed to give a talk on, would have been mostly about dogs and their domestication, and then how kind of unusual cancer. We lost some information about that event. But this one is going to be quite different, but still kind of on the theme of cancer transmission. I gave a seminar a couple of months ago now on the biology of cancer cell transmission, and I mentioned what we're going to be talking about today. So this kind of goes in greater depth in one of those studies that I mentioned. Okay, so briefly I'm going to kind of chat a little bit about cancer transmission in humans. So there are lots of interesting examples of cancer transmission in animal populations. As you might know, it's much rarer when this occurs between humans. Then I'm going to talk about a study that came out a couple of months ago by workshop, which is a case of cancer transmission between two twins in utero. And then finally I'm just going to wrap up with some conclusions and questions. Okay, so I thought I'd start quite simply. This talk is going to be really easy to take in because it's the end of this long session. So probably we're familiar with this idea of viewing kind of the clonal evolution of tumor cell populations. So this figure here is quite popular. It's from a paper in 1976 by Peter and Noel. We described this process, and it's sort of like a paradigm, a simple paradigm of how we think cancer cell populations evolve, where we have sort of positive selection acting on some clones within the population driving their clonal expansion, and then we have some of these clones end up with strata, and some of these proliferate, and we kind of have this idea that cancer goes about, or that tumor cells derive from normal cells that have been transformed by sequential kind of acquisition of mutations that allow them to defy the normal constraints that would be placed on cells and to proliferate unchecked. But typically this is a sort of evolutionary dead end. So most cancers are limited to a single host, and they can't be beyond that individual, thankfully. But there are some instances where cancer has kind of got to that final frontier where you can actually metastasize beyond an individual and into an unrelated individual within the same species. And there are even some instances where you can have cancer cells metastasizing between individuals within different species. And it's important to emphasize that this is not caused by a virus, so this is actual transplantation of living cancer cells between different individuals. And that's what's leading to the sort of the progression of the disease within the population. This is meant to represent canine transmissible venereal tumor, which is what I worked on during my PhD, but it's not actually what I'll be talking about today. So one of the things that the talk is kind of thinking about what this scenario might look like in humans. So we already know that there aren't any serially propagated examples of cancer transmission in humans. But it's kind of interesting to think about if there was to be cancer cell transmission, what are the circumstances under which we think or in which we think that might happen. So if we start to think about it, there are two huge barriers to that ever happening. One of these is migration. So there's the actual physical route of transfer of cells between two different individuals. And the other problem is invasion. So how to evade a host immune response. And there are different ways that we can overcome these barriers in the cases where we're thinking about transmission between humans. And I split these into two broad categories, which are kind of divided based on mechanism, we'll call it. So the first is iatrogenic. So that's cases where you have cancer cell transmission. And this is taking place as a result of a medical or an experimental procedure in a laboratory. And as you imagine, most of these are accidental. And then the other example is mammalian effects, which is the kind of natural route of transmission that happens via the placenta. So we're going to go through each of these mechanisms and describe them in a little bit more detail. And the study that I'll describe later on concerns cancer cell transmission via the placenta. So there are a number of known instances where cancer cells have been transmitted between people by this in a sort of a medical setting. So one example is so incidental transfer of cancer cells via blood, so by transplantation of organs or tissues. As you imagine, it's relatively rare that you would develop cancer from these tissues, but that's one scenario. It sort of makes sense when you consider that most of these patients will be immunosuppressed. Then the second example is simple accidental needle stick transfer. There are a couple of case reports of this. There's one really interesting scenario where there's actual metastasis in the patient after the needle stick transfer. And then this last scenario is something that's also actually very rare, but deliberate transfer. So I don't know if anyone would have heard of Chester Sullivan, who's kind of a notorious, or now notorious immunologist from Sloan Kettering. But in the 1960s, he deliberately inoculated patients at the Jewish Clonal disease hospital in Dublin with living cancer cells, experimentally as a without consent, to see what would happen. But that's the only, well, there are a couple, but that's the most infamous example of deliberate transfer. But it's not really clear that any of these kind of quirkier examples of cancer cell transmission actually tell us anything interesting about biology. And I think the route that's much more interesting is the placenta, and there are a couple of instances where we see this happen. So one is one of the diseases that my group works on now, which is gestational trophoblastic disease. I don't know if anyone will have heard of it before, but it's a rare placental tumor type where the tumor is made up of placental material. And often the tumor itself is made from a totally aberrant placental material, so it would be completely androgenetic, meaning that the tumor is only made up of paternal DNA. So it isn't even a semi-allographic anymore, it's truly allogeneic. Then there are a couple of known examples where there have been transmission of cancer cells between the mother to fetus in utero. And then some examples that I'll talk about in more detail, where you have transmission between monozygotic twins in utero. And it kind of makes sense if you think about, what's going on in the placenta. So in terms of its architecture and in the immunology, it's perfect setting for cancer cell transmission between individuals. In particular in humans, where we have this extremely invasive type of placenta, so we have a hemocorial placenta, where you have maximal contact between the fetal and maternal cells, which means that you can have cells moving across this, or cells in contact within this space. There's also kind of one other route which isn't described here, and so far I can only find one case report, but it's where you have transmission from the cancer within the fetus into the maternal blood circulation. And I'll go into one case of that. So this is from a review that was written on cancer cell transmission by Melville. So you kind of see maybe a large handful of cases of this actually happening. And most of these are melanomas, and they're sort of sporadic, non-linked cases. This is one particular example that I thought would be maybe more interesting to point out. So this was a study in, recently enough, in the New England Medicine. And it was 2 cases of lung cancer, which had derived from uterine cervical tumors in the mother. So the idea was that probably there were cervical tumor cells circulating in the fluid in the birth canal, and that these were aspirated into the lungs, and later led to lung tumors that were detected in these infants. And this was just picked up by a totally routine screening or sequencing of matched tumor normal sequences where they found that there was this mismatch between them and identify this tumor. So I think they're building this out a little more now, kind of looking for specific examples of this. This is a case report I mentioned that was really quite interesting and pretty unusual of cancer that had originated in the fetus, and then the cancer cells were detected in the maternal circulation. Even though it isn't described in this report, so this is based on correspondence with the authors of this publication. The case itself is also really interesting to people, I don't know, the people who spoke earlier about clonal hematopoiesis and cancers here, but it was a mother who had suffered from arachnophobia, quite severe arachnophobia since childhood. So 2 months prior to becoming pregnant, she had been using aerosolized permethrin within her apartment to rid it of any insects. But they think it was showed that it was likely this exposure to permethrin that led to this case of congenital leukemia. So this is a preterm female baby who was born with leukemia, which derived from this permethrin exposure. And this is just a scenario where they took EB173 cell lines. So these are cells derived from a 45-year-old male with a chronic myeloid leukemia. It's quite common to use it. But they basically incubated this with permethrin to show that it leads to a very characteristic a cleavage or a translocation on the MLL gene, which is KMT2A. And essentially they're able to also pick up on cells showing this characteristic translocation in the maternal circulation. But the mother was able to clear these cells after a couple of weeks. So there was transmission of these this cancer cell clone across the placenta into the maternal circulation. Thankfully she was able to clear it. And I should also say permethrin is a super common insecticide. It's widely used. It's also used as a medicine, so topical treatment for lice disease. And then the other scenario I mentioned is that you might have, so as well as having contact between the mother and the fetus, you also have contact between twins in utero. So this is an example of monochorionic twins. So this is where twins would share a placenta. So about 75% of monozygotic twins share a placenta. And there are a couple now of quite nice case reports demonstrating in utero origin or developmental origin of pediatric blood cancers. So this is one example from a paper in PNAS with 2 twin pairs where they're able to demonstrate developmental timing of the mutations leading to ALL. So the study that I'm going to describe throughout the rest of the talk is reasonably similar in terms of this kind of setup. So I mentioned that there were already a couple of case reports showing that you could demonstrate developmental timing, but so far, or until this paper that we published, it has only really been shown for pediatric cases. But it is unclear whether you might have an in utero transfusion between twins that would persist until adulthood. so they could have this really, really long latency after transmission event showing up. So these are two of my colleagues at the work has carried out. So this is Adam Reid, who leads a hematology group in Oxford, and this is Nicolas Sousos, who is a MD PhD student who's working on this. So basically in 2019 they identified this really quite interesting family where for the rest of the talk I'm going to call them twin A and twin B. So basically twin A showed up with a diagnosis of primary myelofibrosis, which is a type of myeloproliferative neoplasm, doctors call it MPN. But at age 37, and they're able to establish that this was caused by quite a typical recurrent mutation in the calreticulin gene, using kind of a genotyping panel. And then a year later, this twin B showed up with almost exactly the same disease. And with the same causative mutation, so they showed that it was the same 52 base pair deletion in CAL1. So that led them to wonder whether or not these twins had both, well, one twin had given this disease to the other, which would be quite surprising because it would mean that there was a latency of 35 years from when this cancer could likely emerged to when we had managed to detect it. So I'm just going to talk through, I guess, whether it did originate in utero, but kind of talking through the logic of how we work that out and some of the implications of that, I guess. So obviously, at that point, we wanted to show that this definitely wasn't some germline event that had happened. To give you some background on the twins, so we had established that they hadn't undergone any sort of transfusion, they had normal BMI, they didn't smoke, they didn't drink, they remained in the same household until 25 years of age. They didn't have any other autoimmune or chronic medical condition. And they had no history of blood cancer or MPN. Similarly, they had no history of exposure to ionizing radiation or any sort of chemical or occupational exposure. We couldn't work out whether they had actually shared the placenta, so it's more than likely that they did because of the statistics and monocytic. But that's what we were able to establish. So basically by fragment analysis, we just looked at whether or not we could pick up this CAL4 mutation, which is the cancer-causing mutation, in their purified T cells, in dermal fibroblasts, and in granulocytes. So if it had been acquired somatically, we'd only see it in the granulocytes, and that we're not going to see it in T cells or fibroblasts, which is what is shown here. And basically we just saw very, very low levels in T cells, but this is enough to convince us that it was somatic. So then the next question is whether they might have, okay, for some reason part of this is missing, even though it's on the slide. Okay, I can just talk you through what should be here. Yeah, so the next question was whether or not they had both independently acquired exactly the same event, or whether or not we could show that there had been some common origin. So what it's supposed to show here is we basically wanted to do a type of lineage tracing. So we thought that if they had acquired this CAL4 mutation in utero, then it's likely that they probably had other shared SNP mutations aside from just this cancer-causing or likely cancer-causing mutation. And that these shared mutations would have occurred at around this in utero transfusion point, so this point here. So we just wanted to look at the sort of the mutation burden across both of the lineages, and we also wanted to look at the different mutational signatures, so that would give us some idea about what was driving the acquisition of these mutations and whether or not these processes were similar in twins. Essentially what this is supposed to show is that the burden in both twins was extremely similar, and would make sense if there was if this had come from a somatic event in utero. One of the other things that we wanted to do to kind of links in with the title, so this pre-cancerous evolution, is that we wanted to look at the clonal architecture of the cancer, which is sort of what we're doing here. So this CCF is a cancer cell fraction, so it's basically where you take the variable allele frequency and you sort of correct it for the sample purity at the copy number. So that you could say something about the proportion or the number of cancer cells that are being affected by a mutation in a more precise way. And in doing this, we sort of were able to infer various things about the kind of early clonal evolution of the tumors and the later subclonal evolution. One interesting thing is this TEC2 frameshift mutation, which is subclonal, and it's only found in twin A, which has a slightly more aggressive phenotype than the other twin. But the main advantage of doing this is that we're able to build a model. So we've made a sort of Bayesian pulsar model based on these observations to give us an idea of whether or not we could say something about what was the time frame here. So if we could say, okay, we model the number of mutations that we think are occurring from birth to diagnosis in both of the twins, which we can get at here. And we can also say something about how many of these, what proportion of these are clonal versus subclonal. So basically, in inferring the pre-cancerous evolution, we use this model to say how many mutations that occurred up to the point where there's a transfusion. And luckily, the model told us that this event it also supported this in vitro origin. So then looking at these different mutations in each of the twins, we also just want to look at what might be driving those. So you'll probably be familiar already with this idea of mutational signatures, where you can take mutations occurring, you can look at the context, so the bases occurring on either side of that specific point mutation, and then you can say something about the pattern of these mutation types, and then you can link these with various exposures. So here we have, this is actually quite, I mean, if you are familiar with signatures, you'll know that this is sort of expected. So most of the mutations in both twins are caused by these endogenous clock-like processes. So SBS1 and SBS5 are just typical signatures that you'll see in both. So you see that they almost have exactly the same processes driving the somatic mutations that we identified in both of the twins. And the only difference that we can really see is in the indel mutations. So we see that there's some difference in the processes that are driving indels in both twins, but we didn't really have time to dig into what this might be. And also it's just a very limited case report, so we only have these two individuals. And it wasn't sure it gave us any more interesting information about the biology. But potentially this is also linked with the later phenotype. And then one of the other things we wanted to do is, so we have this kind of novel situation where we can look at maybe what is the cell origin from which in which this cancer-causing clone originated. So what we did is we just took the cells and then sorted them, index sorted them to single cells and genotyped colonies made from those single cells. So essentially, so this and this is kind of flow cytometries showing us breaking up the cells into HSPCs or heteropoietic stegomorphic precursor cells. So basically, what we would expect from this is that if we were looking at the cell origin of these HSPCs, that we'd be able to identify the Clonal mutation, which is this driver, in HSPCs for both of the twins, which is what we did find. And then interestingly, we also found and we also would expect to be able to see all of the shared mutations that we identified in HSPCs for both of the twins. That makes sense. And there's this one interesting situation where we have a shared mutation present in one of the colonies that's wild type for the Clonal mutation, which tells us that this shared mutation probably occurred before the Clonal mutation. It also gives us some idea that maybe it was twin A in which this cancer first originated, and then this was transmitted by the placenta to twin B. This sort of feeds into this, but basically so this is us kind of genotyping the index sorted colonies. But the take home is that the precursors of both of these traits is likely linked with kind of canonical HSPC hierarchies. I feel like I'm flying through this, but hopefully it's all kind of making sense. So essentially the take home from all of this is all of the evidence, including the modeling we did of pre-cancerous evolution, points towards this one transfusion event which occurred later on in gestation and in utero. And then we have this hugely long disease latency, which is largely driven by, or largely subclonal evolution occurred during that time. And that the drivers that were required later on in cancer evolution didn't really lead to any change in the eventual phenotype that was seen in the twins. So then we sort of wondered if we can see this really low latency in CALR positive blood cancers, but if this is something that can also be seen in other types of blood cancer that are derived from different driver mutations. So one of the candidates we wanted to look at was JAK2 positive mutations. And this is kind of something we're thinking about building out to a larger study, but we sort of included it in this paper almost like a proof of principle. So we were able to pull data for 3 individuals that we knew were positive for JAK2V 16-7F. So basically, we knew they had eventually developed a disease with this linked with this mutation. So we wondered if we could go back to their neonatal blood spots, and if we could sequence these, if we could identify that mutation in the blood spot, which would show that it had been acquired in utero, and that you could see this same type of disease latency coming from different blood cancer drivers. So in the end, we were actually only able to pull this data for 3 patients. Largely, it's because all these blood spots were contained almost like in an airport hangar of boxes for you to go through manually pull the data, so it took a really long time to do this. And this is mostly done by mucos who spent a lot of time trying to get these cards. But essentially, out of the 3 individuals that we looked at, for one of those individuals, we were able to show that this JAK2 event had probably in the utero. We were able to detect it in the Guthrie part at about around 1.34% or something. But this was really interesting to us, and we think it would be kind of a neat way to use these cards that are allowed to go unused. And the other thing is that this is sort of fits in quite nicely with really rapidly expanding volume literature, looking at these really long latency periods, particularly in blood cancers. So this is from quite a nice review that was published by Clonal and Cell. But it's describing the findings from a recent paper in Nature by Yoshi Nangalia and Peter Campbell, who are based at the Science Institute of Cambridge. But essentially what they did is that they whole genome sequenced single cell colonies for about 10 different individuals with MPN. I think in total it was around maybe over 950 whole genome sequences. But essentially they're able to get these extremely detailed phylogenies of clonal hematopoiesis and show exactly what we did in this study. So that there are individuals, I think in the study, found someone with a JAK2 positive mutation, a really 54-year latency period during which they had got kind of symptom free. But it kind of gets at a question that patients with the disease want to know about as well, which is how have I had this for what's going on in this disease, but it's kind of tied itself with this. I guess also, so the study of cancer transmission is directly sort of quirky, and a lot of these findings like this, which I find quite interesting, are sort of collateral damage from looking into something where you are really just trying to think about cancer cell transmission and not so much that is underlying biology. But this raises lots of interesting questions. So I guess I mentioned that typically it had been thought to be exclusive to childhood disease, but we're now seeing these long labels periods in adults. But it also has important implications for our ability to make early diagnoses and interventions. But also, fetal origin in some MPNs might present kind of unanticipated opportunities for different therapies. It also leads us to question whether or not this time of origin, so whether or not these events are occurring in utero or postnatal, leads to different phenotypes of disease. And it might be important that we kind of segregate those patients so that we can work out whether or not there is a linked phenotype. Also, I think the most interesting question to come from that is whether there's something about fetal hematopoiesis or the fetal environment that makes it particularly permissive for acquiring these types of mutations. So the study that I mentioned way at the very beginning about permethrin, which was this insecticide, but the authors think that under normal circumstances, while this exposure might not lead to a type of cancer-causing translocation, that fetal hematopoietic cells were more vulnerable than most cell types to this type of damage. Okay, I think I've brought to say half of what I actually meant to say. Yeah, so basically, kind of in my group, we're looking at different instances of cancer cell transmission. So still looking at those transmissible, some of those transmissible cancers in animals, also looking at placental disease. We've also started looking at syncytins, for anyone who's interested in retrotransposons and their activity in placental tissue. I just thought I'd include this kind of nice shot of Dublin because some people mentioned that they hadn't actually been to Ireland. It's quite nice when it's not raining. And yeah, if you have any questions about this, because I mean you can read through it, but do email me or tweet at me. Yeah. Okay, thank you. Any questions?
Amazing talk. I would dare to say also really great science. It's not that you can amplify yourself as in the incubator, but most of us are in the primary street. I would maybe a reason that I would like to know more use. Can you speculate actually that these mutations can be caused by some epigenetic factor, or is there a possibility to look at the inheritance of this constitution?
I mean, yeah, that would be really interesting. I guess I should have kind of made that disclaimer when we're really only looking at one part of the story. But I think that would be a really kind of interesting next step to take.
I don't know, is it technically, is it possible to say that the samples are limited, let's say, with this?
Yeah.
But it would be maybe interesting to.
Yeah, absolutely. It would be really interesting.
Thank you.
Are there any questions?
So I have a very naive question, maybe. So in this case of a cancer transmission that you were showing at the beginning, wouldn't that create like a rejection from the host towards the cancer cell, like preventing the development of the tumor, or?
So I guess that's a big question. One, so we have the idea that maybe kind of these transmissible cancers are arising possibly more often than we think. But it might happen that you have cancer transmission happening once, but it isn't able to make it to the next individual. It's kind of extinguished, where that's the end of the plot. So it could be this happens a lot of different animal populations. Or even that there have been transmissible cancers in the past that have then died out in this way. But then for some species, so one I didn't mention is the Tasmanian devil, because they might be familiar with the facial tumor. Maybe some species are more vulnerable to these kind of diseases taking hold than others, like potentially related to the immune system or levels of inbreeding. One thing that you do see, so I mentioned this canine transmissible venereal tumor, which we know was established thousands of years ago and has been kind of propagating as a lineage since then. But in some dogs, you do see spontaneous progression. of the disease, so they develop a tumor. Sometimes it can get quite large, and then will then be able to mount an immune response spontaneously, and the cancer completely survives. So I guess it's kind of both linked to the individual and the species, how successful these lineages are. If that answers your question.
Yeah, I mean, more or less,
Yeah.
it's an open question.
So I might have one question.
Yeah.
Do you know maybe what's the incidence rates of cancer in twins in children in like early ages?
I don't know what the answer is to that, but so there are a couple of things we wanted to do after this, and when I proposed it right for this talk, I expected that we would have started some of those already. So one of those is looking at some of the kind of twin sequencing studies that are available to see if we could kind of happen to come across images like this that maybe you can kind of pass over. But I should look at what the actual rates are for twins.
I guess it's not going to be that high, so it's going to be hard to.
Yeah.
To okay. Are there any more questions?
Oh,
Yeah, I have a question. What most problem missed this, but like very genetic cancers are also described in terms of this, the, for example, homozygous loss of functions in BRCA. Was it excluded? Or not only BRCA, but any of the electrical libraries?
In this case.
Yeah.
Yeah, so we kind of looked at any other potentially.
Everything, even like
Structural
structural variants, everything, expression was fine, everything was fine.
Well, okay, so we didn't actually, so in terms of genome level effects, we looked at everything.
Okay, we looked at everything.
Yeah, we didn't do any additional sequencing beyond that.
Karina?
I don't know if it's a polling question or like a couple but obviously you cannot amplify your sample like me, and it's very hard to study because of that. So I'm wondering if there are a lot of also in affairs where just one twin got cancer, one is still in utero, and then the other, like you can trace it back. So you get the whole shit back to the utero, and then the other didn't. And then try to get like a negative evidence kind of.
So as in showing that there was, so they're totally divergent phenotypes, so basically one
Yes, exactly.
phenotype, one was a, they have a drug. Yeah, I mean, I haven't seen anything, so I've seen some studies on twins in very old age, where they show that they have drivers that would typically be associated with choromanophysis that are in both twins, but they don't have the genetic disease. But it's the same phenotype that twins because they don't have disease. But we haven't actually looked at some maybe that's something we could do with this twin data to work that out. Yeah.
Yes, that concludes the question. So thank you very much. I actually am going to give you an answer because that's not a question. But in case you can hear it, if you want to twist it the way that. Okay. Thank you very much.